Assalamualaikum everyone, welcome back to this channel. This is me Aniveed and you are on the channel of Medicare. So as you know that we have started the lectures of pathology, and in every lecture we are discussing acute inflammation and it's all the things. And as you know that in the previous lecture we have discussed the first component of acute inflammation, which was the vascular changes. We have seen how the vasodilation is occur. First the transient vasoconstriction occurs, then the vasodilation occurs, and endothelial cells activate, and all of these processes occur, and the vascular permeability increases. Transudation process occurs, and how the vascular permeability increase by the vasoconstriction of the endothelial cells, which is done by some mediators like the cytokines, TNF alpha, and interleukin, histamine, some other mediators like leukotrienes and bradykinin. So these mediators are contracting the endothelial cells and increasing the vascular permeability. And the second way to increase the vascular permeability is to increase the process of transcytosis. Trans mean transportation and cytosis mean any cell or any protein. If we increase the process of transportation of proteins, then the process of vascular permeability or there will be increase in the vascular permeability. So these are the two steps by which we can increase our vascular permeability. So now we have covered the first part of our acute inflammation. Now we are going to start the second component of our acute inflammation, which was the cellular events. So the word cellular events mean some kind of cellular changes will occur that will regulate your acute inflammation. And the major cell that involves all in the inflammation is your leukocytes or your neutrophils, you can say it, because leukocyte is your white blood cell. So the major cell that play important role in the inflammation is your white blood cell. You can say it white blood cell, you can say it leukocytes. One thing. Okay, some students they misunderstand. They understand that the leukotrienes or the lymphocytes and the leukocytes are the same thing. They are not the same. Lymphocytes, B cells or T cells, they are the type of white blood cell. But the leukocytes and the white blood cells, they are the one thing. You can call it white blood cell or you can call it leukocytes. And lymphocyte, it is a type of white blood cell, like the neutrophils, okay, like the macrophages, monocytes, all these cells, they are the types of white blood cells. So in the cellular events, there the major important role is of white blood cells, or you can say it leukocytes. Only changes occur in this leukocyte. They can undergo the process of migration. They will migrate into your body, and the second word that used for the migration terminology is the recruitment. Recruit man, they mean the same. Migration and recruitment both meaning are the same, is that your white blood cell migrate at the site of injury. And the second important step of cellular events is the white blood cell, or you can say leukocytes activation. They will activate because their activation is very necessary to kill that microbe and to heal that injury site and to prevent the further exposure of the microbe with it. So that's why the activation of white blood cell is very important. So these two are the major steps of your cellular events, or the major step of acute inflammation. So your white blood cell, what is their function? They will travel at the site of the injury where the injury is occurring, and they will ingest, they will phagocytize that harmful agent, that offending agent, that pathogen, that infection causing agent, and it will remove all the dead tissues, tissue damage, or the or if the tissue damage become prolonged, then it will become chronic. But the white blood cell kill that microbe, and if it has not the capability to destroy that microorganism, then the microorganism will remain and it destroy the tissues, and the inflammation will become prolonged, that lead to the chronic inflammation. But white blood cells are the major role to kill the microbes. So for the first process is of the leukocytes migration. So we are starting how the white blood cells migrate. What are the mechanism of it? So white blood cells migration, or you can say recruitment, same thing. So how this all the process is occurring and how the steps it are. So this step of leukocyte migration is given as its name is the leukocytes recruitment. It is a multi-step process, and it is the manager step. So it is going to manage all the things. So this is not a single step process. It has multiple steps it has, and then the leukocyte migrate at the site of inflammation. So what step it has? It has the four steps. So the first step of leukocyte migration is you can say margination, or you can say the rolling. Okay, so this is the first step. Second step is the film adhesion. We will discuss all of these steps in detail. Third step is transmigration, and diapedesis. And the fourth step is the chemotaxis. These four steps combined, and they will do one thing, which is the white blood cell or the leukocyte migration at the site of inflammation. So first we will discuss how the white blood cell marginate or roll. So this is the first step. So first is the margination. These two terms are the different. Margination is something else, rolling is something else. We will discuss it separately. Okay, so the first thing is the margination. So we will discuss how the margination is going to occur. So as the blood flows, the leukocytes move to the edges of the vessel. Okay, so first step of margination. This is not migration. This is margination. So you have to remember the difference between the margination and the migration. So migration, first step is margination. So as this blood is flows, and you have the inflammation, then what will occur? Some lymphocytes, which are the part of white blood cell. I am drawing a lymphocyte at that side. So this is a lymphocyte cell. So they are present at the center of your blood vessel, like that. So they are present at the center, and they move toward the peripheral wall of your blood vessel, like that. They will adhere, they try to adhere with the wall of your blood vessel. This process is called margination. And why this kind of movement is occurring? Due to three major reasons. First reason is the stasis, due to reduced blood flow, you can say the accumulation of the blood cells. Second reason is the chemotaxis, and the third reason is the cytokines. I have said the cytokines is the major component of inflammation. Its knowledge will repeat again and again and again. It covers in every topic, so that's why you have to keep in mind. So why this movement occurs at the peripheral part? Why the migration occur? Due to stasis, your blood cells accumulate and reduce their blood flow. That's why they move toward the peripheral part. Sometimes chemotaxis occur. Like for example, this is my skin. and at this site the injury will occur. So this site is injured, so it sends signals at that part that allow the lymphocytes to move toward the peripheral part. Okay, and also due to some cytokines these kind of movements, so they became to move along the wall of your blood vessel like this. Okay, not at the center. This is the process of margination. And the second process is the rolling. So the second major process is your rolling. That's very simple, that your leukocytes roll with the wall of your blood vessel, so they will roll along with it and move forward. Okay, so what is the reason behind it, why it is, and what kind of mediator is doing that? So the reason behind the margination is these three reasons: stasis, chemotaxis, and cytokines. Now the reason behind the rolling, why it is rolling along the wall of the blood vessel, due to one reason is the cytokines. Cytokines is the major reason, as I have said. Every reason is the cytokines behind it. So what is the cytokines they are doing? Actually they are activating the endothelial cells, and they expresses the selectins are the adhesion molecules on the endothelial cells and the leukocytes, and that mediate the rolling process. So अब आपके पास क्या करते हैं कि लाइक मैं एक और ब्लड वेसल यहाँ पर बना देती हूँ for it, so that you can observe how the rolling is occurring. So actually your white blood cell has epithelium layer here. Am I right? Yes. So what is going to occur here, because of injury the cytokines release, and these cytokines allow your this lymphocyte to adhere with it and to roll along it, because it activates the endothelial cell. It will go activate over all the endothelial cells, and some kind of adhesion molecule will activate on the endothelial cell. So normally it is non-active form, but now it become activated due to cytokines, and it will allow the lymphocyte to bind with it. So now our this lymphocyte bind with the selectin, and it adhere with it. And this selectin is not only one molecule. After some time another selectin molecules came, and this lymphocyte roll and transfer to this site. Then we have next selectin molecule here, then lymphocyte roll and bind to it. So this is not a frame attachment, this is the temporary attachment, and in this process the lymphocyte rolls, and it occurs with the wall. I have drawn it at the center. It occurs along the wall, like now you can observe it clearly, that how it is occurring along the wall. It is rolling along the wall due to endothelial cell activation through the cytokines, which expresses selectin molecules, which are the special adhesion molecules that allow the adhesion of lymphocyte through that area. And when it adhere, it will start the lymphocyte to roll. So this was the major key behind the first step of white blood cell migration. So we have covered the first step of white blood cell migration, which is margination and rolling. Margination is the movement of white blood cell along the wall of the blood vessel due to steasis, chemotaxis, and cytokines. And rolling is occurring due to cytokines activation of the endothelial cells, causing the selectin molecules to adhere, are the adhesion molecules, and allow the adhesion of lymphocytes to the wall of blood vessels. Now we are moving to uh, okay. So some additional information in the rolling is that our leukocyte selectins. This is the loose attachment. Leukocyte attachment to the selectin. This is the loose one, not the frame attachment. And the selectin has some types. Okay. So for example, selectins have the three types: selectin E, selectin P, and selectin L. E selectin, P selectin, and L selectin. E selectin is also called CD62E. This is called CD62P. This is called CD62L. ठीक है, बिल्कुल सेम चीज़ है. सॉरी, तो ये नाम है CD62. आगे F, ई की जगह आपने एफ ई लगाना है. सॉरी, ई. CD62E, CD62P, CD62L. ठीक है, ये भी इन्हें नाम लिए जाते हैं. सो अगर कोई आपसे पूछ रहे व्हाट इज CD62E, तो आपको पता हो दिस इज द ई selectin adhere molecules present on the endothelial cells that are activated by cytokines during the rolling of white blood cells, and this is a step of cellular events or white blood cell migration. So your E selectin or your P selectins, they are present on the endothelial cells mostly, but your L selectin, this is mostly present on the lymph node, lymph vessels or the leukocytes. So their location is, but mostly E selectin or P selectin is present in your endothelial cell. So this selectin molecule is, for example, if this is E selectin, next selectin molecules will be P selectin. Next can be is L selectin if it is a lymph vessel. If it's not a lymph vessel, then it will be E selectin. Okay, so in this process the leukocytes move or roll along the wall of your blood vessel. Okay, so after discussing the first step of white blood cell migration, which is migration and rolling, second step is the frame adhesion. Now, after the temporarily attachment, we move to the frame adhesion, permanent attachment, and how it is gonna to occur. Let's discuss it. Okay, so now we are gonna to frame adhesion of leukocytes to the endothelial cells. So this is your blood vessel. These are your endothelial cells. First, they were temporarily attached to the selectins molecule, E selectin, P selectin, or L selectin. Now we don't want this selectin. We want the permanent attachment of this leukocyte with the endothelial cell. And who is responsible? Who is connecting both of them? The major thing that is causing the frame adhesion between them is the integrins. This is another component that involved in it. In rolling, we have selectin. In frame adhesion, we have integrins that play important roles. So these integrins, where they are present on the surface of your white blood cells. So white blood cell के ऊपर ही होंगे, यानी इस lymphocyte के ऊपर ही ये integrins अटैच होंगे और ये जाके endothelial cell के ऊपर अटैच होगा. It is present in the inactivated form. It converts into the activated form due to the conformational changes that is done by cytokines. And integrins ligands, they are present on the endothelial cell. For example, ICAM1 proteins and VCAM1 proteins. These proteins are present on the endothelial cells. This is the major thing. Now I am going to draw the diagram here clearly so that you can see it. This is a blood vessel endothelial layer. First we have the selectin which attach the lymphocyte. Now this lymphocyte also contain attachment. Now we change its shape so that you can remember the shape of selectin and the other things. This is integrins attach onto the surface of lymphocyte. On our endothelial cells, we also have a ligand which is called a integrin ligand. This is integrin ligand and this is integrin. Now integrin will go and attach to this ligand, so it will go and attach with it like this. Okay, and this called the frame adhesion. Now it is firmly adhere this lymphocyte with the endothelial cell. Now the frame adhesion occurs between both of them. And the types, or you can say the example of the integrant ligand, they are the two proteins here. First is the ICAM one protein and second is the CAM1 protein. So these two proteins present on the endothelial cells, they are acting as integrant ligand, and your integrant normally it is inactive form, but it is activated due to cytokines. So it means cytokines have their role in film adhesion also. They have also the role in the margination, they have also the role in the rolling process, and they have also the involvement in the second step, which is the free median. They are causing conformational change and they are changing it. But the major component and the major protein that you have to discuss in the free median is the integrants, and the major protein that you will discuss in the case of rolling is the selectin. So these are the small little things that are very important for the viva point of view. Similarly, these two proteins which are acting as a integrant ligand, they are also very important for the viva point of view, so you have to keep them in your mind before going toward the viva question. Okay, so now we have covered the first two steps of your white blood cells migration. Margination, rolling is done, primedian is done. Third step is transmigration, or you can say it as diapedesis. Now we are gonna to see it. Okay, guys, so the third step is the transmigration and diabetes. So, what is going to occur? So, I am making the diagram to make it easy for you to understand what is occurring in that step. So, as you we have say in the previous step that the endothelial cell is there. On the endothelial cell, we have integrant ligand, which can be ICAM one protein and VCAM one protein, and the ligand present on the lymphocyte cell, it firmly attach the leukocyte with the endothelial cell and making the frame attachment, and the cytokines involves in this area because cytokines cause the conformation change, and due to which this integrant activates and they perform their action. Now in the transmigration process, there will be activation of another protein, another channel, you can say a protein that acts as a channel. So this is a protein which is normally it's transportation is closed here. So normally this gap is closed, but it helps our leukocytes to migrate outside. So we have to detach this lymphocyte, and we have to make this lymphocytes to move outside. So normally this gap is closed, it doesn't allow lymphocytes to move outside. But when we have any injury outside, like we have the injured area is this is my skin and my injured area is that, so we want lymphocytes to move from this side to that side because we have to do lymphocytes migration. So this is the migration done by this protein, which open its gap and allow the lymphocyte cells to move outside at the site of the injury to kill the microbe, to do the phagocytosis or all the things that its responsibility is to perform it. And this protein is called, it is abbreviated as PECAM1 protein, also be called CD31. CD31 name is very popular. They can said to you what is the role of CD in the case of acute inflammation. So you should know that CD31 or the PECAM1 protein, this is the protein that allow the transmigration of, or you can say diapedesis of lymphocyte outside into the extravascular spaces so that it can travel toward the site of the injury and kill the microbe at that site, kill the dead cell at that site, and protect our body from this microbe. So this is the major component. And the so we have covered the first four steps of white blood cell migration: margination, rolling, film adhesion, transmigration, or diabetes. Now the fourth step is the chemotaxis. Now let's see what is chemotaxis and what things are occurring in it. Okay, guys, now our lymphocytes chemotaxis will occur. as we have seen in the previous topic that the lymphocyte, it will moves out with a protein called PECAM1 protein with CD31, which allow this lymphocyte to move outside to travel toward the site of the injury. Now let's assume that the site of injury level is too far, like my injury site is this is my injury site. So now lymphocyte has to travel this area. Okay, now chemotaxis or chemokines help with this movement. So chemotaxis is the movement of lymphocytes to the site of the injury due to some chemicals, due to some signals, and the signals or the chemicals are chemokines. What the chemokines are doing, there are some chemical signals that are released from the injury site. So this injured site release the chemokines that allow the lymphocyte to travel to the site of injury. So they bind to the peptidoglycan and help the leukocytes to move. So how they move, the cytoskeleton microfibrils, they attach themselves to the leukocyte, forming the pseudopods, and that allow it to travel toward the site of injury. So क्या होता है, इसके साथ अटैच होते हैं साइटोस्केलेटन माइक्रोफाइबर्स जो कि इसके pseudopods बना है. फॉर एग्जांपल ये इसका pseudopods बन जाते हैं इस तरह के. जैसे आपने अमीबा को देखा होगा, उसके इस तरह के pseudopods होते हैं. ठीक है, वो इजीली इनके ज़रिए से ट्रैवल कर लेता है. बिल्कुल इसी तरह के pseudopods बन जाते हैं लिम्फोसाइट के ऊपर, जिसकी वजह से वो इजीली ट्रैवल करके इस साइट पे पहुंच जाता है. दिस इज़ द मेजर पर्पस ऑफ chemotaxis. Okay, after that in your book some important note is written that after the inflammation within the first 6 to 24 hours, the neutrophils, first of all, neutrophil is the first cell that travels toward the site of the injury within 6 to 24 hours. Why? What is the reason why other blood vessels don't travel? Because most abundant lymphocytes in the blood is a neutrophil. So it's mean in the acute inflammation. In the acute inflammation, we are talking about the neutrophils. Neutrophils, because they are quick in response, they show the initial response, and they are most abundant type of the white blood cell. That's why they can immediately go toward the site of the injury and performing the action there. And after the 6 to 24 hours of the inflammation, the neutrophils undergo the process of phagocytosis and they die. So due to this process, because of the most of the neutrophils died in this process, because of the germ, they have himself destroy themselves, so they die, and their number or their amount particularly decrease, and the amount of monocytes, which are the second most abundant white blood cells, they begin to work. So after the neutrophil, its second abundant cell, which are the monocytes, they will activate and they will show their action. So in the case of acute inflammation, you cannot say that all the major role is performed by the neutrophils. No, neutrophils can decrease in the number because they are dying, they are fighting and they are dying. When they die and their amount decrease, you will stand with another army, which is the monocytes. They will also start the fight against that germ, against that villain. So this is the thing that I want to. And there is some exceptional case. This is the case neutrophils work when we have a bacterial infection, when some bacteria, some fungi, protozoa act on it. Now when protozoa act, eosinophils are there, but when we have bacterial infection, mostly neutrophils play important role. And when we have any viral infection due to any virus, lymphocytes are the most abundant leukocyte that travel here. Now we have some case. This is a case that why in the case of pseudomonas infection, neutrophils stay in the infiltrate for several days, because Pseudomonas aeruginosa. This is a bacteria. It is very resilient pathogen. It produces the toxin and produce the biofilm around it, and that's why it is very toxic or it is very strong. So that's why neutrophils continuously recirculated at the site of the infection. Usually in other infections, neutrophils stay for a day or 2 days, and after that they will die. But in the case of Pseudomonas aeruginosa, it is very powerful bacteria. It play the biofilm around it, and it produces a toxin. That's why it is the resilient bacteria. It is silently killing you, and also silently killing the neutrophils, whose stay is for a day or for the 2 days only. After that they will die. But in the case of pseudomonas, they will travel continuously toward the site of the injury, and they are causing these things. So guys, we have covered the topic of white blood cells migration or recruitment. In the next lecture we will see how the white blood cell activate. So this was all about our today's lecture. I hope you like this video. If you like it, then please subscribe to my channel and press the bell icon so that you can get the notification of next upcoming videos. So take care. Allah Hafiz.